I realise with this slide, I'm excluding probably 90% of the audience with the cultural reference, apart from some that are sitting on the row at the back. That's okay, Gareth's already put his hand up. This is Eamon Andrews. He used to run a show called This Is Your Life. Eamon Andrews is not here to recount this, so I'll do it myself. For those of you that think that's a bit too narcissistic, you should know that at the squash club, where I spend far too much of my time, according to my wife, I'm the only person in the club known to say, good shot, to myself. So I think I can do this. So we'll start from the beginning. I grew up in a place called Wellingborough. Many of you will not have heard of it. It doesn't have any particularly endearing features. As you can see, as one of the main photos, I chose the Arndale Centre, which is replicated across many cities and small towns from the South Midlands up to the north. But it does have some iconic cultural pieces. Does anyone recognise that symbol at the bottom? Bauhaus. Well done. There's always one. Okay, so the Bauhaus was an artistic movement started in Germany, unfortunately, not in Wellingborough. But it did lend its name to the group next to it, which is also called Bauhaus. It was formed in Wellingborough and is the pioneer of Gothic, rock, and post-punk, if you're into that sort of thing. I'm not, but they're culturally important, and it's about the only time I can say that for Wellingborough. The other one is in the bottom right, and I'm sure more people are going to know this. This is the most decorated English footballer ever.
It's goals.
No. Anyone? Oh, Mark's floundering. No. Phil Neal. 4 European Cups, about 9 league titles, and the rest of it. And he's from a suburb of Wellingborough. Okay. So it didn't have a lot to offer, but I am proud to come from there. One of its best features is that notwithstanding it's right in the middle of the Midlands, and I just see Mike Coleman there, he's also from the environs of Wellingborough. It's culturally diverse, particularly for a small town of that size. It's had a lot of Indian, Pakistani, Caribbean, Italian, and Polish in the 60s, 70s, and 50s as well. Okay, so it's multicultural. And that, in a sense, also shaped me. That's my mum there on the left. That's my dad. It's one of those 4. Can anyone guess which one is my dad? Yeah. The one on the left. The other bald one. Oh, there's another one. Anyway, those 4 are all my uncles. Mine's the one on the left in the dodgy pinstripe suit. And I chose this picture deliberately because it always reminds me of this. The scene from Only Fools and Horses on the Jolly Boys outing. In the top right there is my stepfather, and in the bottom is my sister with my mum again, her 3 kids, and my daughter. So it's just photographic evidence that they do sometimes come to Liverpool, and they enjoy it, as I do. Okay, so skipping from there, we'll skip all the way through when I had hair. I used to wear Joe Bloggs jeans. And I went to university. I went to Imperial College in London in 1993. I brought the stereo deck with me. It gave me great comfort at times, but overall I really enjoyed Imperial College. It was a great place to be. It was a bit too science-focused, to be honest. If I had my time again, I'd probably enjoy being a student in Liverpool more than there. But it was a great place to learn. At the end of my studies, I did a final year project, which is quite intense at Imperial. It's all focused into about 10 weeks, and it's full-time. And sometimes it's, several hours a day past normal working hours. I did that in the lab of Professor Andrea Crisanti, who at the time was more of a parasitologist, actually. And we were working on looking at knocking out some key genes that might be important for the invasion of the parasite into the liver cells in the human. At the conclusion of my final year project, I'd obviously done all right because he approached me in the corridor, literally ambushed me and said, listen, I've got a PhD. You've got a week to decide. It's now or never. One time only offer. You need to let me know within a week. So there were 2 problems with that, or 3 actually, but the main one was that I hadn't actively sought a PhD. I was considering it, And when you're flattered by being offered a PhD, it's a very strong, powerful force, and it's hard to say no. The second thing was that it was on Cryptosporidium parvum, which is a pretty boring parasite in my view. And the scope of the project was just to catalogue different Cryptosporidium isolates from public water bodies. So for me, that was about as interesting as watching paint dry. Notwithstanding that, it was hard to say no to the offer. And I think this is relevant to younger students, and I can see some of the Emirates students in here. You should really make sure that it's an active choice. And that's what I did. So I said no. I left. I came back in 1998. I told Andrea that it wasn't the right time. I wanted it to be an active choice. And I wanted to, I'm paraphrasing myself here, find myself in Africa and see what the real situation was on the ground in Africa. So with that excuse, I left temporarily. I wrote a letter to the MRC in the Gambia, in fountain pen, sent it off in the snail mail, in the normal post, waited for a reply and went back to Wellingborough. And I waited, and I waited. And I did loads of temporary jobs in the meantime. Some in labs, some litter picking, even around my old school, which was interesting because some people still recognize me. And I waited for this reply, and the reply came back, again, in fountain pen. Sorry, we don't have any spaces. And so that was about 5 months later, at which point I was ready and decided to have a look around actively for PhDs. I applied to the University of Glasgow, to what was then the Wellcome Institute of Parasitology. They had a doctoral training program similar to ours, and it was really great. They offered me an attractive position, actually working on Thyleria, not that it's relevant. But also to have a choice, I also approached Andrea again to call his bluff, to see whether that 1 week only offer that was now or never was actually now or never, or something different. And of course, he was bluffing. He had a very attractive project, which was to try and modify the mosquito genome in ways that could alter its vectorial capacity. So I was betwixt and between these two choices. And it was very difficult for me to decide. Glasgow was a new place. It was a dedicated institute of parasitology. It had a doctoral training program, whereas Andrea's lab was more of a free spirit in Imperial College, which was something I knew, but it was also quite anarchic and competitive at the time, sometimes in good ways. So I had a choice to make. And I was in a full dilemma. And actually, it was my stepfather, when he heard the conditions of the two offers, that said to me, the offer at Glasgow at that time, even though it was a welcome PhD, the stipend was £6,250 for the year. And his words were, that's a disgrace. That's slave labour. At your stage, you've done your degree, you've done your classification. They shouldn't be expecting you to live off that. And I followed his advice, so I went back to Imperial. And I started again with Andrea on a project that was to design a genetic transformation technology for vectors of malaria. This was exciting for me. It tapped into some of the skill sets I developed in my project. It was back on malaria, which is what I wanted to do. But this time it was on the vector. And it was in collaboration also with Fotis Kafatos, since departed, who at the time was the Director General of the European Molecular Biology Laboratories. Okay, so why do we want to, at that time, develop a genetic transformation technology for the mosquito? This will not be news to some of you, but I deliberately wanted people from professional services and other non-scientists to come to this audience. So you won't mind me repeating some of the facts that you already know, many of you at least in the audience. Malaria is the world's deadliest vector-borne disease. The mosquitoes that transmit it, we call them vectors because they vector the parasite from one individual to another. It's only transmitted by the bite of female mosquitoes and only of a particular genus, which is Anopheles. In 2021, there were nearly 250 million cases of malaria and over 500,000 deaths. And nearly all of these are occurring in children, and nearly all of those are occurring in sub-Saharan Africa. And an important feature of malaria is that although there are several thousand species of mosquitoes, it's really only a handful of them that are responsible for nearly all of the transmission. So a mosquito bites you, it takes up the parasite develops in its midgut. After about 10 days or so, those parasites have become infectious to the sporozoites and to the salivary glands. And once the mosquito takes another blood meal, once it's got salivary gland sporozoites, it ingests a few salivary gland sporozoites, and the transmission is complete to another human. So there are potentially different places we can attack the malaria parasite. In the human, in the vector, the mosquito, or by preventing contact between the vector and the human. So targeting the parasite means antimalarial drugs or vaccines. Targeting the mosquito vector means residual spraying of insecticides within houses where humans and mosquitoes cohabit. Removing breeding habitats for mosquitoes. Mosquitoes lay eggs in stagnant water bodies, so removing of those where they're proximal to human habitation can be a very effective way of breaking the transmission cycle. And as many in this audience will know, the use of insecticide-treated bed nets, which both act as a barrier to prevent contact between the human and the vector, but when there is attempted contact by a mosquito, it comes to the bed net and is killed or at least repelled by the presence of the insecticide. And over time, since the Second World War, where malaria has been removed from several areas where it was previously endemic, particularly in Europe and on the peripheries of Africa, it has been targeting the mosquito vector that has had the most tractable gains in reducing malaria transmission. So that's what makes an understanding of the mosquito biology really interesting to us, and did at the time, because many of the traits that make it such a good vector, such as how long it lives, such as where it likes to feed and who it likes to feed on, how well the parasite develops within the mosquito vector, and how it seeks out mates and reproduces, all of these have strong genetic components. And that's where this concept of functional genetics comes in, which is making changes to genes or genomes in order to determine the function and the effect of those changes. And so the Holy Grail really is making genetic modifications that allow us to understand some of these key phenotypes, such as resistance to the parasite, insecticide resistance, changes to its behavior, how it reproduces, and how mosquito sex is determined, for example. So the concept is to have this genetic change that you want to make, you produce that in the laboratory through DNA cloning, and you get that into the mosquito somehow. So what is the minimum unit that we need to get into the mosquito? Well, this is about the only molecular biology you're going to get in this. So for those that are molecular biologists, don't worry. The basic design of a transgene is that it has a promoter, which is a switch which responds to certain transcription factors that may be present in some cells or at some time in the mosquito development and not in others. And therefore, the gene of interest, which is downstream of that switch, is only turned on or off in certain cells or at certain times in mosquito development. And this gene of interest, which is DNA, produces a transcript when that gene is turned on. That transcript is like a message. It's called RNA, and that RNA, the message, is converted into a protein, usually. And it's that protein that will have some dominant effect on some traits, be it transmission blocking in our case, be it altered behavior, whatever we might like. And the best analogy for this that I've seen is like a kitchen, where the DNA is the cookbook, the RNA is a quickly scrawled copy of a recipe that's taken from that cookbook. Then you take that to the chef and they mix together all the amino acids, which together make a protein. And that, although the pioneers of molecular biology would kill me, is basically the central dogma. Okay, and so with that, we can potentially then understand the genetics that are essential for mosquito capacity to support transmission, which is fine. But the ultimate promise is this idea of genetic control. It's having those mosquitoes with altered genetic characteristics and releasing them in order to have some effect on a population, either to suppress that population or to get that genetic trait that you've modified to increase in frequency in the population and over time replace the existing genotypes that are there. Because this relies on mating of those released mosquitoes with the wild-type mosquitoes, they do the hard job of finding niches that can be very difficult for humans to reach. Again, because it relies on mating, it's species-specific, therefore non-toxic. So I haven't actually told you how we might get that genetic modification from the lab bench to the mosquito. And that, at the time, when I was doing the PhD, was the big question mark. And what we and others were all hurrying to try and do was to use this class of element, which are called transposable elements. These are mobile genetic elements. They're effectively genomic parasites. They're very simple in structure. They have two inverted repeats, so sequences that repeat here and here, but inverted on each other. And the only thing they encode is one gene, which is a transposase. And this gene is an enzyme that is able to recognize these inverted repeats and cut the gene out of the genome. This mobile piece of DNA can then go and insert somewhere else. So they can hop around genomes, basically, and that's why they're called jumping genes. You have these in your body. All insects have these in your body, but different in their bodies, but different classes. And by this jumping around, they can increase their number, their copy number in the genome and overall in the population. And so the idea was to try and use these genomic parasites, basically cut out their guts, which is the motor that actually gets them from the original location, and replace this with a transgene of interest. And on another component, have just the transposase gene that's not able to act on itself because there aren't these inverted repeats, but it can only act what we call in trans on this molecule. So it can excise this molecule and insert it from the plasmid into the mosquito chromosome. That's the idea. But this is quite a rare event. So the other thing that we need is some sort of flag or marker in order to recognize when this element has come from here and inserted into the mosquito gene. We need all that to happen inside the germline of the mosquito. And the only way to get to the germline of the mosquito is to look at the embryo and try and inject these components into the very early embryo, where these progenitor cells that go on to form the germline are initially formed and reside at the pole end of the embryos. And so the idea is that we can try and get that transposition event to occur in one or some of those cells that will then go on to form the germline. So they will either be precursors of the sperm or the eggs that the mosquito makes as an adult. And if it's happened in some sperm, those sperm, you can see the little green ones down there, when they go and fertilize a wild-type mosquito, all the offspring will have a copy of that genetic element. And we'll be able to identify those individuals as transgenic because they will have received a modified chromosome and hopefully are marked and flagged, as we said. So we designed a methodological approach that was pretty sound. We first had to identify what would be a suitable transposable element that might be able to work in the mosquito. We had to develop a way to inject Anopheles mosquito embryos. And we had to develop a way to show and quantify how active these were in the embryo in order to see that they might actually work in transforming germline cells. And we needed a dominant genetic marker to identify these rare transgenics, so that flag I was talking about. The issue at the time was we didn't have any of these, neither a suitable transposable element, neither a way to inject mosquito embryos, nor to test activity in the embryo, nor a dominant marker. So we reasoned that we would take a transposable element that one of us or our collaborators had worked with quite extensively and had been shown to be mobile in its host genome, but also in other insect genomes when you transfer it. And the element we focused on was Minos, which you might not be surprised to know was developed by researchers in the island of Crete. Minos was their local god. Having decided on that transposable element system, we then had to adapt a microinjection system to the mosquito embryo. This is a system that is ordinarily used, you'll have seen this on the BBC and the rest of it, for in vitro fertilization. Normally it has two arms, it's an inverted microscope, one here to hold the human embryo, the other one to inject it with a needle. So we took off one arm and we adapted it for the injection of mosquito embryos. Now, as many in the lab, particularly in the front row, will know, this is a tricky procedure. It takes a lot of time to learn, takes a lot of skill and dexterity to do properly, and you also need to have exactly the right timing on the mosquito embryos. You have about a 2-hour window in which these are suitable. I call it the Cinderella window, in which these are suitable to be injected. So many can die, it's challenging technically, and it takes a lot of dexterity and resilience. So it can be demotivating when you're trying to learn that. And I know that from personal experience. I've never told the lab this. I know that from personal experience because at one point, before we'd made any of these, Andrea, who'd spent about £25,000 on this setup, came out with this classic quote, that this was the most expensive paperweight in history. Because no one was bloody injecting. Anyway, we got over that barrier. We got injections working. We got a dominant marker that we could show was working well, at least injecting in Drosophila, but it was also working well in the mosquito. And a key finding was that we could mobilize our transposable elements from one plasmid into another. And we could select these plasmids and show that they showed this canonical transposition event that was really mediated by that transposon, because the footprints it left on the plasmid it escaped from and the plasmid it inserted into were perfectly in line with how this transposable element should move. So we had all the components together. At that point, it was just a numbers game and going through and injecting the mosquitoes. And eventually, we got there, and we were able to transform for the first time these Anopheles stephensi mosquitoes. And you can see here we have a wild-type individual. Here we have one with 2 copies of the transposable element inserted, and here with 1 copy of the transposable element. And they're all very precisely integrated. So at the time, this was a big breakthrough. It was reproducible. It was the first time we'd had a malaria mosquito transformed. And it got us quite some publicity. And another gratuitous shot of me here with hair. And you can see the perils of genetic engineering, as you can see how long my arm is. For the record, that is actually Andrea's arm. I don't want the story to run away with itself about genetic engineering. Okay. So at that point, I had some credit, some clout, and once you start to produce results, as my lab will know, it's more likely you get sent to conferences and get a bit of visibility. And that's what happened. I went to Strasbourg in 2000, and this was, apart from being a lovely city, this was part of a research training network. And this is not going to be a Brexit rant, but to say that this research training network was, I would say, the epitome of how the European Commission can work. It was led by Fotis Kafatos. It had Jules Hoffman in it, who went on to get a Nobel Prize. It had Babis Savakis, it had Andrea Crisanti, Bruno Arca. Loads of prominent insect biologists that were at the top of their game and were genuinely committed to this thing that they called mobility of researchers. Everyone moved around. You'll see what I'm getting at in a minute. And that's where I first met Gareth Lysett. He was there. He was sharing a room with me, weren't you?
I remember that.
Yes, you do. Remember, because we got locked out. And it's where also Gareth was helping in being matchmaker between me and the future Miss Citizenship Award 2025. She was there, frantically worrying about her slides. Nothing's changed. And yeah, so this was the first announcement that this was working, and it started a load of collaborations between us, Jules Hoffman, that was looking at all these insect innate immunity genes, and for the first time we were able to put these genes into mosquitoes and see their real effect. So there were loads of traits that could now be altered genetically, which was great. But there's one thing altering a mosquito genetically in the lab, even if we assume we can make that either resistant to the parasite or have some behavioral change or some change in its reproductive capacity. The real elephant in the room was, how do you ever make that into a population control tool, i.e. how do you get that genetic transformation into a population in a meaningful time frame? And that's the challenge, because the number of mosquitoes that you're ever able to rear realistically is only a small fraction. These are the modified mosquitoes. There's only ever a fraction of the wild type population here in black. Okay? And so your initial release frequency will be relatively low, and that is not expected to change generation upon generation. So if it's low at the start, it will be low at the end. And the truth is that many of these modifications are unlikely to increase mosquito fitness. In fact, many times they will slightly decrease mosquito fitness. So they're certainly not likely to be selected. And so over time, they're likely to be lost. So that was an issue, but it was one I knew about, and I'd completed my PhD. I'd done well, I'd done what I set out to do. But this was an issue that was a challenge for me. I couldn't see at that time how to turn that into a population control tool. And so it got me thinking. This is also about a year or two after I'd met future Miss Citizen 2025. And so I had my own personal motivations for leaving the lab as well. And something that interested me was something that was coming out at the time, was a really interesting phenomenon that had been found in a commercial application of transgenesis. So these are petunias, I think. Is it petunias? Yes. Good. Okay. So these and other flowers are prioritized or desirable because of this strong color pigmentation that they have. And the genes that encode the proteins that are essential for the production of that pigment are known. And so the idea was, okay, maybe we can make these even more pretty, even more powerful in terms of their color performance. When they put genes in, they saw the exact opposite. It would start to become white in different areas. And also, if you get viruses that have a piece of that genome in them, they also make the flower white. So when you see these variegated white flowers, usually it's because there's a virus in there and something's going on. And so this got me asking, is this a possibility not just for these extra transgenes that are put in there, but is it a possibility for transposons? This is a general mechanism that happens to all repetitive DNA elements. And so I combined two things. One, a need to go to Rome for personal reasons, and two, an excellent lab that was actually working in that field particularly, and that was the lab of Carlo Cogoni. And so in late 2002, I went there. That is Rome, by the way. Very pretty, very exciting place to be. What's probably less exciting on the surface of it is the organism that I was working on, which is this pink bread mold. The scientific name is Neurospora crassa, but from humble beginnings, we can get some really amazing discoveries. So actually, this pink bread mold, Neurospora crassa, was the organism of choice for the study of many genetic phenomena, and it led to the original postulation that one gene encodes one protein, just as I showed you, actually, ironically, in that transgene design. And that got these two guys the Nobel Prize. And any of you who have done A-level biology, or certainly undergraduate degree biology, you will remember these things which show the segregation during cell division. That was a long time ago, not when I went, when they did that. But since then, it's continued, or at least did, to be a treasure trove of different genetic phenomena that relate to this idea of silencing of repetitive sequences. So without going into its life cycle, it has a haploid phase and a diploid phase. So, and through all of those three phases that it has, it's always doing something actively to silence or mutate repetitive DNA sequences. So when it goes through sexual reproduction, it has this thing called repeat-induced point mutation. So any sequence that's repeated in the genome will be mutated. Nucleotides will just be changed, and the sequence will just be jumbled. Just prior to meiosis, any gene that's got an extra copy of itself will be silenced. And the same thing, but a different phenomenon, a different mechanism leads to silencing also in the asexual stage, which are these pink media here. And this lab that I went to really was on a shoestring budget. And that's why I called it the Rumpelstiltskin lab. They were basically spinning straw into gold in that lab. They had a perfect organism, and there was this new phenomenon of RNA interference. They knew that RNA interference was active in this organism, but because of the handle they had on genetics, they were able to identify how copies in the genome were initially recognized as being different from all the other genes. They recognized how does it produce an RNA that is somehow different, and how does that RNA get recognized for degradation, which is this idea of RNA interference. So they had 2 or 3 science papers, a couple of Nature papers, and it was a great place to be, but they were really working on a shoestring. So I came in on a European grant and was paid about 3 times more than all the other staff in the lab. So I was initially public enemy #1, but I got over that, and I had a great time there. I spent nearly 4 years there, and I made a couple of important publications. One was showing that transposons are subjected to a certain type of this silencing, so that was relevant to the work I'd done in the mosquito. And the other thing that I did was recognizing how these repetitive elements are recognized initially as being aberrant and different. So these were good for my career, but the biggest thing I think I learned in Italy was an expression that stuck with me, which is this, which is tutto il mondo è un paese, which is that literally translates as the whole world is a village. But in Paul McCartney languages, people are the same wherever you go. And that was true. You know, the challenges affecting PhD students, the challenges affecting students leaving university, and the challenges affecting postdocs were even more stark there. So I think sometimes it's easy to forget that we are very lucky here in the opportunities we have to do research and to build a career in this. There you have excellent students leaving university that don't really have job opportunities within the research sector in academia. Anyway, I finished there, and it was time to come back to the UK for a number of different reasons. And Andrea Crisanti kept phoning me all the time I was there. And this time I could speak Italian, so I really understood the idiosyncrasies of what he was saying. And he kept trying to get me to come back. And again, I used the excuse that it's never good to go back, using the football analogy. Anyone know who that is?
Howard Kendall.
Well done, Denise. Howard Kendall was probably Everton's most prolific manager. When he went back, it didn't work out too well. And that was my worry, whether the magic would survive a second act. So instead of going back, despite his protestations, and having this new project, which I didn't really want to look into, I went to UCL for a while, and I worked on worms, and particularly this phenomenon of aging, where it says aging at UCL. Although I was aging quite rapidly, I was working on the phenomenon and the genetic basis for it. And it's quite a remarkable organism and phenomenon that single mutations in a single gene can sometimes have extraordinarily strong effects on lifespan. So this DAF2 gene, for example, can double or triple the average lifespan of a worm, and in combination with another mutation, it can make you live make the worm live 5 or 6 times longer. So there's some really interesting genetic phenomena there, but I just didn't get on with aging, and I didn't particularly like working in that field. And at the same time, there'd been some moves in the mosquito field, some of them led by work that was done here. What was being obvious was that, again, this concept of targeting the vector and it being the most effective form of malaria control was being shown in real time. Around the turn of the millennium, there were several large different types of intervention measures introduced, and malaria fell from about 1 million deaths in 2000 to about 400,000 in 2015. And when you work back what the different effect of the different interventions was in taking those malaria deaths down from the counterfactual rate, i.e. what would have happened in the absence of any intervention, we can see that the majority of effect came from treated mosquito nets and indoor spraying of residual insecticide. So it really boosted this idea that vector control is our most effective form of malaria. But at the same time, again, a lot of work done here in this institute was showing that notwithstanding these gains, insecticide resistance was on the horizon and was rapidly proliferating across Africa. And we know this is slightly disingenuous because this is describing time after it came. But we now know that some of those gains are stalling, if not retreating. So we definitely need new insecticides. Ideally, we need new drugs and vaccines. But it really forced home this idea that we need novel vector control approaches that are complementary and synergistic to those that are proven. So that was one thing. The other thing was that, to my conundrum previously, this concept of gene drive had been introduced. A gene drive, the definition of gene drive is that any genetic element that's able to bias its own inheritance among offspring. And this was the pet love of an evolutionary biologist called Austin Burt, who was at Imperial College at the time, not long in actually, and he had read about the work that I had done with Andrea Crisanti, and he had hooked up with Andrea Crisanti to try and make gene drive a thing for mosquitoes. So I went back, had a little chat, played football quite a bit before deciding to join with Nicky Winbickler, who was one of the postdocs leading some of the molecular biology. And this mantra of never go back turned into, oh, go on then, give it a go. And so this concept of gene drive, how does it change the landscape? So this was the previous paradigm where all those modifications over time would be lost. The reason that the frequency, i.e. the number of insects that have it in one generation and the next doesn't change, is simple Mendelian laws of inheritance. So mosquitoes, just like you and I, have 2 copies of every chromosome. So when you have a copy of your genetic trait of interest, this mosquito will mate with this mosquito. Each mosquito contributes only 1 copy of the 2 chromosomes. You as humans do exactly the same. So it can either contribute this chromosome or this chromosome. So there's a 50% chance of this chromosome being included with the trait, or a 50% chance of the wild-type chromosome. In this individual, it can only contribute a wild-type chromosome, so it's 100%. So when this fertilizes this, half these will have the genetic trait, the other half won't have the genetic trait. And so one had it, one didn't. One had it, one didn't. The frequency doesn't change from one generation to the next. Okay? That's very important. And so we have to somehow get around those Mendelian laws of inheritance. And that's where this concept of the gene drive comes in. It biases the inheritance of either that chromosome or that specific genetic locus among the gametes so that it can be close to 100% of gametes containing that trait. So now, starting from the same starting frequency, when an individual mates with a wild-type mosquito, nearly all the offspring have it. When these mate in the next generation with a wild-type mosquito, nearly all the offspring have it. And it starts to increase in frequency, and so on, such that the trait can go rapidly to fixation. And that is, in essence, the concept of a gene drive. So they are, or at least can be, designed to be very invasive. You start from an initial low frequency in a population, and rapidly, over time, that population is transformed. And if there's connectivity between different neighboring populations, it can spread to those populations at differing rates, depending on how connected they are, depending if there are any hidden reproductive isolation barriers, or whether there are geographical or ecological separation barriers. So that's the principle of a gene drive. How do we actually build one? And again, it was Austin that had been looking examples of gene drives in nature. So these things, this phenomenon exists in nature. And a very simple class of gene drive is something called homing endonuclease genes. Again, just like those transposable elements, they're very simple. They encode just one gene, but this gene inserts on a chromosome inside its own recognition sequence. And every time it comes into contact with a chromosome that has the wild type sequence, this endonuclease gene, so this DNA cutting enzyme, recognizes this specific sequence, cuts that chromosome, and we get a broken chromosome. This broken chromosome has to repair itself. It can either just cut and shut, or better, it uses this intact chromosome as a template where the sequences match, and it reads across the intervenous sequence and copies it into this region here. So by that copying mechanism, after the cutting, we now have both chromosomes containing the original homing endonuclease gene. And so now, whether you get this chromosome in the offspring or this chromosome in the offspring, they all have this gene. How am I doing for time, by the way? Nearly there. Okay. So this was the concept, but then along came this system called CRISPR. And essentially, this does something very similar. It's a very specific DNA cutting enzyme that recognizes very specific DNA sequences. But the beauty of this is that the cutting is determined by the provision of this small molecule, which is called a guide RNA. So by changing the guide RNA that you provide to this setup, you can cut any sequence that you want in the genome. So it's effectively like a find and replace for genomes. And so we reprogrammed these, since they're so similar to these homing endonuclease genes, to work exactly as a homing endonuclease gene. And this was the work of Drew Hammond that came to me initially as a BSc student and then stayed on for PhD. So we were reconfiguring these CRISPR-Cas9 elements to work as selfish genetic elements and mimic exactly how homing endonuclease gene works by putting them in specific places in the mosquito genome and asking them to copy across and see if they could bias their inheritance in the germline. Okay, so that's the way that they copy. There are two ways that you can actually have an effect on the population. You can either cause them to copy at a genomic region that is essential for the mosquito, or you can drive with them a copy of a gene that is actually doing some effect against the parasite. So the first example would be an example of population suppression, and the second example would be population modification. This is what we've worked on. And the principle is this, that you release the gene drive at low frequency. It does this copying and therefore starts to increase in frequency in the population without any effect. But what you're actually spreading is a recessive lethal mutation, so that when it starts to increase in frequency and you get individuals that have both copies of the gene drive, they have no functioning copy of the gene that you're targeting, and therefore they're all non-viable. So as the gene drive increases in frequency, population fitness is drastically reduced. And we showed that despite this targeting of endogenous mosquito genes, you can cause these to rapidly increase in frequency in the population. These worked, but they weren't optimal. So then we moved on to different genetic targets. And one of the key targets that we've been working on, and this was the idea of Andrea Crisanti, and I must say I didn't actually think it would work at the time, was to target this gene that's involved in sexual determination. So it's a single gene, but the transcripts, the messages that come from the gene, are different in males and females, and they make slightly different proteins. So if you target only a region that's contained in the female transcript, then what you've got effectively is a kill switch that only works in females. And so when you drive that through the population and you have both copies of this double sex disrupted, you only start to see an interesting phenotype in the females. And so they start to have characteristics that more resemble males, such as their plumose antennae, maxillary palps, and the presence of claspers. Importantly, these are non-biting and they're non-fertile. So as you spread the gene drive through the population, starting from an initial low frequency, it rapidly goes to fixation. And because you don't have any viable females, the egg output is quashed. And therefore, you can get total population collapse. We see that in small cages. We see that also in large cages that we've done in collaboration with Polo GGB in Italy. Now, these somewhat more mimic more ecologically relevant conditions, such as swarming markers, oviposition behavior, and host seeking. And again, we see the exact same result. This gene drive initially released at low frequency goes to fixation. And this type of experiment allows us to make predictions of what these might look over time. And so this is just showing that over a large geographical scale, you might get from a single release quite drastic impacts over time, even 4 years later. And this is what led to an expansion of the program that I was working on at the time, which was called Target Malaria, into a very large program. And I just want to make the point here, I've only got time to talk about some of the molecular biology and the genetics, but this is a huge operation, and it's not just genetics. We've got modeling, fieldwork, community engagement, training, risk assessment, and all the rest of it. And through the development of this program and the opportunities that allowed me, it's given me a huge and valuable network for outreach, training, how to think about designing pathways to translation, and some of the work has also had influence on policy and practice. And for this, I want to give a shout out to Dr. Karen Logan, who's the senior project manager there at Target Malaria. And she saw in me a talent for engaging across different sectors and explaining some of this science, even if I might not be doing the best job of it now. And she gave me these opportunities, and for that I'm very, very grateful, because they have led to further things, which I'll talk about soon. And I'm also very grateful to Stephanie James at the Foundation for National Institute of Health, who involved me very early on how to develop consensus pathways for testing of these gene-drive modified mosquitoes and interacting with regulatory authorities. And I'm also thankful to Valentino Cisnetto, who was a trusted aide across a lot of this interaction and a really great travel partner. With this, as I said, I'd allude to some of these, it's given me an opportunity to help cultivate the next generation of scientists. And I was just looking, I've done this for years, some of this interaction with PAMCA, we've done gene drive training courses and all the rest of it. And I came across this photo, and I think it's from 3 or 4 years ago. And this is Gregoire Savadogo. Is he in the audience?
Yeah.
There he is, yeah. So where was this? Kigali, it says there, Kigali.
Kigali.
Yeah, so we met at Kigali at this course, and then now here he is in the lab. So this outreach, these training opportunities really do come back to roost for the good in the long term. And Federica Bernardini, shout out to her also, that's been helping me over the last few years with some of this. And that's the same thing that motivates the thing that Martin alluded to, which is the development of this MRes in tropical health and infectious disease research. Hands up if you've in the MRes or done the MRes. Yeah, great to see you here. Solomon? Did I see Solomon somewhere? No.
Solomon's sick.
Okay, all right. So not just those young researchers, it's allowed me to build a lot of ongoing collaborations. And again, Martin mentioned a couple of them. Jonathan Kayunda, Uganda Virus Research Institute, Abdoulaye Diabate in Burkina Faso, as well as Mamadou Koulibaly, who unfortunately passed away suddenly, relatively recently, but his legacy still remains, and he set up an African Centre for Molecular Engineering in Mali. And through my latter period there at Target Malaria, I also set up some ad hoc collaborations with Charles Wanji that have led to fully funded opportunities. And we're good partners here at LSTM. And all this is important because, rightly, the funding landscape is totally shifting. Funders, this is just the Gates Foundation, but it's something that's being replicated across all the major funders. They are now starting to prioritize the building of local capacity around these technologies that are either complex or new. And so for genetic technology, this is very, very important. Here in 2008, you can see that nearly all the funding was in Northern America and parts of Europe. And now in 2022, you can see it's much more dispersed, and there's huge swathes of it in LMICs around the equator. So this is something to get ahead of. This is where the travel is heading, and it's where we need to be. How long? 5. Yeah. Okay. This is brief. Does anyone know what that molecule is?
DDT.
Huh?
DDT.
Well done, Zach. You'd have passed the PhD. This is DDT. I wouldn't know it now that it's DDT, other than I looked it up to put it here. So I came for a viva. It was Adriana Adolfi, and it was a great viva. But a rumor got out that I'd asked her to draw me the structure of DDT. I want to put on record that is not true. Hillary, as you would if you were future Dean of Research Culture and Integrity, came straight to me and said, "Is that true? You asked Adriana to draw the structure of DDT?" I said, "Absolutely not." So I want to put that on record. But that encounter did lead then to a conversation once she'd calmed down and realised that I hadn't asked to draw the structure of DDT, about actually coming here. And so I did, actually, basically. Within a year, I'd moved. I thought it was time to get a new change of scenery and take new opportunities and new adventures. And so in 2019, January 2019, I came. And since then, we have continued in some of the work we're doing on gene drive, but we're also diversifying. We're moving into new vector species, new genetic targets, and applying some of the genome editing technologies to tractable problems like insecticide resistance, as well as capacity building with partners. And I just wanted to say thank you to Hilary Ranson initially for recruiting me again. This is her more relaxed pose. This is Martin. I think he's escaping from a risk register meeting when he previously had a department. He's doing everything he can to get out. So I won't spend long on this. It's just to say that if you want to get a real handle and really push down malaria control, these are the handful of species that I referred to earlier. These are within the species complex, so they're closely related. But this one, Anopheles funestus, is drastically different and yet a very dominant malaria vector. But it's very challenging to work with. And so just a shout out to the guys at Light. They know I'm one of their biggest fans. So when I came here, I saw a strategic opportunity to make the most of the school's unique selling, what does it stand for, the P there? Point. Unique selling point. And one of these was the provision of these world-class insectaries, but also the provision of this GLP-accredited mosquito lines, where possible. And Funestus is hard to rear, so we were able to piggyback off their production of Funestus originally and translate some of these tools to work well in Funestus, and they do. The other thing that we're doing is looking at the genetic basis of insecticide resistance using some of those genome editing tools and answering what are the genetic changes that matter in a mosquito that appears to be resistant to pyrethroids, which are the active ingredient on nets, and which are just red herrings. So again, we use this CRISPR, and we can see different mutations that might appear to be under selection in the wild, and then we can, one by one, introduce them and really have a look at what their effect is in controlled environments within the laboratory on standard genetic backgrounds, and see how much they contribute to insecticide resistance, whether they improve overall fitness or lifespan or fertility, or whether they're just red herrings. And we've shown that we can do that. I'm the first ones to do that here. We worked in collaboration with Linda Grigoraki, and she can make some of these classical alleles onto susceptible background. And Ash and others in my lab are working on looking at different mutations and their contribution. And the overall holy grail for this is that eventually we can have a sort of genomic surveillance that allows us to capture mosquitoes in the wild, profile them very quickly genetically, and give them a sort of identikit readout and score as to whether they're susceptible or resistant to different insecticides, and operationally change what we deploy as the best vector control. I think I can skip this because Martin's getting agitated. But this is just to make the point again that we are fully engaged in building molecular research capacity in partnership with several partners in Africa. This one is Burkina Faso, and several of the students are here today. And this is built around integrating these researchers into active research projects here that will form part of their PhD, and at the same time building that physical infrastructure at their labs in Burkina. Okay, so in conclusion, and I've written some words for this because I'll botch this otherwise. I should say there is a whole side of this I haven't touched on. There are roadblocks, there are ethical responsibilities, wider questions that these technologies raise, but that wasn't today's focus. Today's focus, although all that stuff matters, today's focus was on how I've got here and the stuff that we've built to make this at least a technical possibility. And the idea that we could one day edit a species to reduce disease is no longer science fiction. I hope I've shown, and that's the thing that I really wanted to do, is that none of this happens in isolation or with a pre-cooked master plan. I wandered, and got there in the end. But it does take some ambition, graft, and luck, but it also takes a lot of teamwork. So here's to more of that. And with that, I can't thank everyone. I've tried to put everyone that's either been in my lab now or associated with it. Everyone else, from all those partners that I've been so lucky to work with, they know who they are. I've referenced some of them. But because it's my inaugural lecture, and even though I am over time, sorry, I do want to have the prerogative of thanking some of the people that don't always get thanked. And those are the people in the non-scientists and other professional staff that never really get on the award slides. And so that's why it was great to see the team that works with me, Claire, Maria, under Sarah, receive the staff award for excellence. I think these are great. And the professional services, HR, research management services, and particularly Claire and Chris, who I asked to come. I see Chris is there. He gets a spot on my board. These guys are really important. And just it's a pleasure for me to say thank you to them here at LSTM and previous colleagues at Imperial that normally don't get on the slides. And the last thing I want to say is to my family. My daughter's not here, so I'll get less emotional now, which is fine. She tells me she's at the job centre for an interview. Let's see how many times she uses that as an excuse. And also thanks to my wife. I know there have been sacrifices made, but there's also been prizes gained on the way, and I'm very grateful to both her and my daughter. And the last cultural reference is the switch, which I had to use to bribe my daughter to come here, because when I told her we were moving to Liverpool, initially she had a total meltdown. And I said, would a switch change things? And she stopped crying immediately. And this is her wearing some of the football tops from some of those visits to both Kenya and Uganda. Okay, sorry I went on a bit. Thank you ever so much.